everyone, and welcome to the latest installment in this whole series on extreme environments on Earth. So as you've seen in previous episodes, and if you haven't, you should see it online on the YouTube channel, they've already explored life under pressure, life in dry conditions, and under low pH conditions. So today I have the pleasure of presenting to you life with a pinch of salt. So I work at the Museum of Natural History in Paris, France, Muséum National d'Histoire Naturelle. So it is a natural history museum, but it's also a university and a research institution. So I'm going to be presenting to you some of the things that have been fascinating our group about halophiles and life in salt and what that means on Earth, as well as the potential for life on other worlds. So with that, let's begin. You might have heard it say that we are all made of stardust. Well, today I would say we are a bit stardust and a little bit sea salt. We're going to look at salts in the universe. The image you're looking at are halite crystals. So this is sodium chloride table salt that we crystallized in our lab. And you can see that little pink color inside some of them. That pink color is actually living microorganisms that are alive inside the salt crystal. And today we're going to talk about why that is, how that is, and what that means for life. First of all, we need a little bit of context. Salts we actually can find throughout the solar system. We know we have them here on Earth. Here's two great examples of salt environments. The top image is sampling I did in Canada, in Saskatchewan, in one of the solar salt terns in Canada, and the bottom is Majorca. We also know that we can find sodium chloride, table salt, on icy moons. There's been evidence of that under the surface of Europa, as well as in the plumes of Enceladus. And there's been evidence of chloride minerals, including halites that have been found around Mars. So we know that salts are found throughout the solar system. And in particular today, I'm talking about sodium chloride salts. But however, life has only been found on one place, Earth. Everything else is just hypothetical. So when we look at extremophiles, we have to use life as we know it as the basis, but then try to dive deeper into what are the principles of life? What are limits of life at a molecular level? Why are there limits of life? And what does that mean for the potential for life on other worlds? On Earth, we know life can not only survive, but it can thrive in salts. So this is a great diagram taken from an article in Marine Drugs that takes a look at the different groups of extremophiles. Different colors mean different kinds of extremes that they can resist. I'm going to draw your attention to the green parts. These are halophiles. So you see sometimes you'll see multiple different colors. That means that certain halophiles from bacteria, from the archaea, are resistant to multiple different kinds of conditions. In particular, we explore halophilic archaea or haloarchaea. These are often the last surviving members in saturated salt conditions, and they are usually some of the more abundant members when we get to saturated salts. But what does it mean to get salty? Why does life need salt? What does salt limit in terms of life? How does it protect life? How does it preserve biosignatures or the signs of life? And could life exist in salts on other planetary bodies? Is that a possibility? That's what we're going to explore next. So first of all, we have to understand what is a microorganism. Essentially a cell, any cell, but particularly unicellular microorganisms. It's a bag of mostly water. It's got lipids, it's a container around it to separate it from external environment. We have nucleic acids that help us with transmitting information and giving instructions for how to build the rest of the cell, proteins for carrying out work in cells. And this is the basic structure. So when we look at survival and the limits of life, we're essentially looking at these molecules. Salts have a unique relationship with these molecules. If we start with water, water is often referred to as the universal solvent. It's what enables the reactions that we know as the basis of biochemistry, everything to produce energy, make molecules, all the processes that we define as living go through water. The advantage of salt is that it can actually keep water liquid under a larger range of temperatures. Now we know this because when it gets too cold, what do? We throw some salt on the sidewalks to keep from slipping. So we have some freezing point depression. We also can have boiling point elevation. Same thing as adding some salt into a pot of water when you're trying to cook. These are things that help us maintain liquid water, which we know is essential for life as we know it. The other things that salts do is help us with essential cell processes. These are things like electrochemical gradients and transporters. We need to be able to transport and regulate which salts, in this case, I mean ions, are on which side of the compartment membrane. And this helps us with the basic processes and bioenergetic processes for life. But salts are also incredibly limiting to life. Life can be limited in a number of different ways. First, you can have limits by osmotic stress. Now, this happens if you're in an environment that has a high salt concentration. What happens then? If we look into a place like Majorca in the solar Saltern areas, we have high concentrations of salts. In a normal cell, this will cause water to go out of the cell, following its gradient, and will actually dry up the cell. It'll end up sort of like going from a grape to a raisin. Some organisms, such as halophilic archaea, combat this by keeping high concentrations of salts, in this case ions, inside their cells. One thing, if you're an astute observer, you'll notice is that the ionic composition inside the cell and outside the cell isn't the same. We've got more sodium outside the cell, and we keep more potassium inside the cells for halophilic archaea. But the concentration is roughly equivalent on either side. This keeps the homeostasis, it keeps the osmotic balance between the inside and the outside of the cell. Other organisms, whether they're eukaryotes, algae, cyanobacteria, they can use other molecules. These are called compatible solutes. These are things like glycerol or trehalose or actamine. Oftentimes, these are molecules that are multi-purpose. They can be used as carbon sources or to help resist against a number of different kinds of stresses. Nature tends to be fairly efficient. So a molecule isn't produced for usually not just for one reason, but for many reasons. The downside of this is that if you have a high concentration of salts inside the cells, it means that if you take, in this case, a halophilic archaea and put it into pure water, water will actually rush into the cell. And the end result is cell lysis. The cell is going to expand because water is going to enter the cell and it's going to explode. We actually exploit this in halophile labs to lyse cells. When we want to break the cells open and extract things like DNA, we can just put the cells into pure water and burst them open. There's a couple of other terms that are important when we look at the effects of salts. So now we're going to get a little bit more into some chemistry and some physics. One of the things that's also an important factor for the limits of life is something called water activity. So water activity, for those of you who prefer it in an equation form, we're looking at the vapor pressure of water in the sample versus the vapor pressure of pure water. Essentially, it means relative humidity. It's how much water is free in solution and how much of it's bound to solutes. The more solutes, the more kinds of molecules you have, whether that happens to be sugars or salts, the more of that water that's bound, you're going to reduce water activity. And as we reduce water activity, life struggles more to maintain all of the biochemical processes it needs to perform. So here's a great example from the lab of John Halsworth. You can take a look more in detail at this article, and I encourage you to do so. They were looking at measures of water activity, and is water activity really limiting for life? And in terms specifically for sodium chloride, is it the most limiting factor? So what you're seeing is a sliding scale of different kinds of water activity. Pure water has a water activity of 1. That means it's pure water. Saturated sodium chloride has a water activity of about 0.75. And the current record holder is a fungus that is at a water, it can still function at a water activity of about 0.585. This is an ongoing area of research in terms of how much free water is necessary to go through the biochemical processes for cell activity. Now, this is the difference between an active cell and something like a spore, which is in an inactive state and doesn't need to go through all these biochemical processes. Another important factor, and this is becoming one of the leading factors that we think is the limiting part of salts: chaotropicity, cosmotropicity. Okay. Big terms. Essentially, what this means is a measure of the tendency of different kinds of solutions to cause disorder in biomolecules. This used to be referred to for things like the Hofmeister series. So you may have heard discussed of different kinds of ions and whether they tend to disrupt or stabilize proteins and other molecules. Now we discuss more in terms of chaotropicity and cosmotropicity. And again, I'll refer you to various labs who've done a lot of good research in this field, John Hallworth's lab and other labs around the world who've looked at this. In general, chaotropic salts are salts that are going to disorder proteins, aggregate proteins, and are going to generally cause things that we think of as negative effects for a cell. Cosmotropic salts are going to be ones that are going to tend to more stabilize, and stability is often thought of as a good thing, but it can go too far. A cell membrane that is too stable can, in fact, lose its permeability, for example. So there's a balance to be had on both of those things, but different kinds of salts have different kinds of effects. And when we look at different salt environments, whether on Earth or on other planets, we have to be very aware of what kind of salt they have, and then what is their effect on the disorder or ordering of biological molecules like proteins. In fact, salts can become so limiting to life that they've become one of the most limiting factors. This is a study that was done several years ago in the lab of Charles Cockell. Jesse Harrison was taking a look at organisms that had been identified under different extreme conditions. And at that time, you'll see this large empty space. If you look at increasing sodium chloride concentrations, different degrees of pH and temperature, there's an area where no organisms were identified. It's this empty zone at high salt concentration, low pH, and high temperature. That combination appears to be limiting for life. The question we need to ask now is, why? At a molecular level, why is that particular combination more limiting to life than, for example, lower temperatures and other different combinations? And that's really, really the state of the art of where we're at now, is trying to determine the why behind these sort of phenomena. This was also observed, screen to mess, there we are, at a Europe Planets site, Dalal, Ethiopia, which is used to study the limits of life. Dalal, Ethiopia, and the Atacama Desert are two areas of our planet that we know that have high salt concentrations. And also we have an incredibly difficult time identifying any sort of signs of life. Detecting microorganisms at all in some of these environments is very, very challenging. There's a great article that's come out of the group of Purificación López Garcia that is examining the limits of life, and with the detection methods that they used, and they used many different techniques to try to detect life, there were still some areas that they were unable to get a clear biosignature. And two factors or combinations of stresses that they identified as being particularly lethal or limiting for the presence of life, rather, were the combination of chaotropicity and low water activity, and the combination of high salt concentrations and low pH. So we're starting to be able to understand now that it's never just one thing at a time, it's multiple things. Salt isn't just limiting, it mixed with other conditions, it can be even more limiting for life. Now we need to understand why. What molecules are damaged by this? Would that be the same sort of effect if life followed a different evolutionary pattern? Another really interesting aspect of salt is that salts can function as a sort of time capsule. Now, I left a question mark, and I'll explain why. Salts are a good place to find preserved biosignatures. We can find preserved biosignatures not only of microorganisms, but their component molecules, as well as organics that aren't actually from a biological origin, but are carbon-based. This is something that we actually know from our human experience. Salts have been used for millennia for preserving skins or for preserving foods. And in fact, this is where a lot of halophiles that we work with in the lab were first isolated, from preserved skins, from high salt fermented sauces, soya sauces, fish sauces. This is where they came from. And so salts have had this ability to preserve microorganisms from the environment. So how does this happen? If we start with a high salt environment, what you're seeing here is a high salt environment that is full of halophiles. That pink color is from their carotenoid pigments, and that functions like a sunscreen for them in a high UV environment. So with this pink color, what happens as we get more heat and conditions get drier? Well, we start to evaporate, and it forms salts like this: sodium chloride, table salt. Within a table salt, you'll see these small inclusions, and they can be inclusions filled with fluid or gases. And inside these fluid inclusions, which are microscopic, we can have a trace of the original brine and the original conditions at the time of evaporation. And this includes microorganisms that can be present. Halites have been found all over the world, different sites, and you can see here in the different color code in red where cells were isolated from such halites, or where they were just able to identify nucleic acids from such halites. Now, these results still are a bit controversial. The reason for that is, how can we be sure that what was detected was from inside the salt crystal and that was from the original evaporation? When we do an evaporation like this in the lab, we can produce salt crystals, and these are ones that were produced in our lab, sodium chloride halite crystals. And if we zoom in, you can see all those little tiny purple squiggly lines. Those are halophilic archaea. So we have a fluid inclusion that's full of these halophilic archaea inside the fluid inclusion. In this case, this is shortly after they have been precipitated in a matter of a couple of months, and they are still alive. There has been other halites that up to millions of years from which other groups have been able to extract DNA or organisms, and they are from the same group, Halobacterium salinarum. They're very closely related to the lab model that we use. Now, this is important to note that the ages that are attached to this, again, are the age of the salt itself. They haven't dated the microorganism. That's a different thing. The reason for that is it's incredibly technically difficult to do. Analytical chemistry and most of the typical biology, microbiology, and biochemistry that we do in the lab isn't made to do it saturated in salt conditions. It tends to jam up most of our instrumentation. It's very difficult to do. So there's a lot of technological development that has to accompany trying to answer some of these basic questions, like how old are the microorganisms that are inside these salt crystals? But this is where we need to start, is by identifying that there are microorganisms associated with halites. So then we have to ask the bigger question. If there are halophiles inside halites, how are they surviving? And this is where I come back to, we need more tools. We're missing some key tools to answer these questions. We need tools that enable us to be able to look inside essentially a closed box, whether that closed box is a salt crystal on Earth, or even a closed box being an experimental platform in space. How do we see inside a closed box in situ? Well, one way we can do this is by using ground-based terrestrial experiments. Now, for this, an approach my lab has used is to take halophilic archaea, good model organisms that we know a lot about. We know their genome, the proteome, we know their structures, their functions, the different molecules. We know so much about this, and this has been from work from labs from all over the world over decades, and been thoroughly studied. This gives us a good basis to be able to start molecular level analyses. So we take these cells, we evaporate them inside halite fluid inclusions. And this work was just very recently published in my lab from the PhD work of Charlie Favreau, who will soon be defending his PhD. So what he was able to do is start with this crystal. So we show with it a schematic of a halite crystal. Here is our salt crystal. We've got some cells in pink inside the fluid inclusions, but there's some that are still on the top of the salt as well. We also have some proteins. It happens when the cells burst open, the cells lyse, and we have proteins and other organic contaminants that are now on the surface of the salt. So we have two technical challenges to overcome. You have to remove all of the organics, all of the biomolecules from the surface of the halite. This is cells and proteins. And then you have to be able to directly extract the molecules only from the fluid inclusions. Desalt them, which is a huge technical challenge, in order to make them compatible with things like mass spectrometry analysis and proteomics analyses. Now, a lot of work has been done leading up to this from other labs that have produced wonderful protocols on how to deactivate cells on the surface. In other words, kill the microorganisms on the surface. They've also been able to remove all contaminating nucleic acids from surface of halite. However, up till now, nobody has attempted to remove all of the proteins as well. So this is where our group decided to attack. So what they decided to do was take a look at comparing liquid cultures of these halophiles, which we know a lot about in molecular level and omics in terms of their protein expression. Put them inside small salt crystals. Why small salt crystals? Because protocols it's much easier to work with a large salt crystal. If you have a halite the size of your fist, it's much easier to surface sterilize. If you dissolve a bit of the salt crystal, you still have enough material to work with. But the challenge is when you have very small salt crystals. And this becomes important with astrobiology because as we prepare for the return of samples from places like Mars, they're going to be small salt crystals. So we need appropriate measures that are going to be able to remove surface contamination from even small halite crystals and enables us to get at what's inside even a small halite crystal. So skipping many steps, and I'll let you read the article to see so we can not get too technical with this. He was able to find a way to remove all traces of, remove the traces of surface contamination of cells and proteins, develop a method of extracting the proteins directly from the fluid inclusions inside the halite. And this was based on protocols that had been worked with before for liquid cultures and now been optimized really for solid halite crystal. Extract proteins, and then through some desalting optimization, which again is, I'm skipping over this, but I want to draw your attention to the fact that this is a very difficult process to desalt samples sufficiently to be compatible with things like mass spectrometry analysis. And then he was able to analyze exactly what was happening to the proteins as they were acclimating to surviving inside salt crystals. So to cut a long story short, what did he find? Turns out that cells that are in what we call stationary phase, at a point in their growth cycle where they're not going through a lot of active cell division, they're more in a maintenance mode. It's very similar whether in liquid cultures or inside the salt crystal. These cells appear to have very low transcription and translation, so they're not producing a lot of RNA or protein. There is still some exchange with their extracellular environment, and we are seeing indirect evidence of metabolism and including an accumulation of toxicity from metabolic byproducts in that closed environment inside the salt crystal. So these are ground-based terrestrial experiments. What we get out of this is now we've got methods. We've got a new toolbox we can use to remove different kinds of organic, what we call contaminants, from even small halite crystals and extract biomolecules from fluid inclusions of halites to perform detailed molecular level analyses that we refer to as omics, proteomics, transcriptomics, et cetera. And I'll let you take a look at the article for all the details to understand the schematic diagram of all the details that's specific to microbiologists. So I don't want to weigh down the geologists and the physicists with that today. The other thing that salts are interesting for is radiation protection. So radiation is not a big concern for us on Earth, but as we look at astrobiology and the difference between Earth and other planets, it becomes a very big concern as we leave our atmosphere. And for that, we need to move to different kinds of experiments. So we can do ground-based experiments for extraterrestrial environments. And some of these have been done by a variety of groups, some that I've participated with and many groups around the world. And what we've been able to show is that organisms like Halobacterium salinarum are very radiation resistant to UVC radiation, gamma radiation, X-rays, heavy ions, and many other kinds of radiation. And being able to look at each type of radiation one at a time enables us to understand how the cells function. Then we can start to combine them in pairs and in groups of different kinds of radiation to understand the cumulative effect on the cells. So you can see that Halobacterium salinarum is very resistant to UVC radiation and to gamma radiation. And there's been omics level studies, detailed level studies looking at their transcriptional translational responses to irradiation to show us the mechanisms of how they survive. And I'll let you look at those articles for reference. One thing I want to show is that we were able to show that the salts that are inside the cell work also as an aid for radiation resistance. This is geology that's protecting biology in a passive geochemistry. So this isn't enzymes, this isn't an active process. And I'll show you how this works. So if we start with the cell of our halophile, so we've got DNA that we need to replicate, we need proteins that function, we've got water, and these organisms are oxygen breathers. So under the conditions we studied, there was oxygen. We need proteins that function to repair any damage to DNA, and the DNA needs to be able to replicate. In the case of ionizing radiation, we have the major effect. Some of it is deposited directly into molecules like DNA, but most of the ionizing radiation actually affects water. The radiolysis of water produces free radical species like hydroxyl radicals, and that's what will actually induce oxidative damages to things like DNA bases. When there are clusters of damage to the DNA bases during the repair of DNA, oxidized DNA bases, during excision repair, this can produce DNA double-stranded breaks in the DNA. The DNA becomes completely fragmented and the cell dies. The other problem that can happen is that we can have electrons that can interact with oxygen, producing superoxide radicals. And these are particularly toxic to proteins, less so to other molecules, but particularly to proteins. So this is what radiation is doing inside of a cell. The proteins are oxidized, they're inactivated, and the cell dies. In the presence of high concentrations of chloride ions in the cells, however, we can actually have an antioxidant effect that will transform things like hydroxyl radicals into less reactive forms that aren't going to damage things like proteins and other molecules in the cell. So DNA is less damaged, proteins are less damaged. The other effect with superoxide is the presence of manganese, Mn, ions inside the cell. And this was first shown by the group of Michael Daly for Deinococcus radiodurans. And he showed that actually protecting proteins from damage is the key to radiation resistance. We previously thought that it was protecting DNA, and it turns out that protecting proteins from damage is more essential. And Halobacterium, like Deinococcus, has a high ratio of manganese to iron. And so the manganese is able to convert superoxide ions into H2O2, which is less damaging for the cells. So here's a series of reactions for those of you who are more interested in the whole series of reactions. And I encourage you to take a look at Michael Daly's paper. And he was able to show in this wonderful review article in Nature Reviews Microbiology that this is a general tendency. The higher the intracellular manganese to iron ratio is in aerobic organisms. This doesn't apply to anaerobes, but aerobic organisms, the more radioresistant they are. So this is how halophiles can be radiation resistant, even on Earth, where they're not naturally going to be exposed to ionizing radiation. It's a byproduct of their adaptation to high salt environments. So the key thing here is that life resists chemistry, not conditions. Other groups have shown that not only does salt inside the cells protects cells from radiation, but salt outside the cell protects them from radiation. The study was done showing that halite can actually reduce the effects of UVC radiation on halophiles inside halite crystals. So now we have protection outside the cell and inside the cell. And this is interesting if we look at it, and even in terms of protection of organic, in this case, I mean carbon-based molecules from a non-biological origin. In the study from Queenie Chan, she was and group, they were able to show that in the Zag and Monaghan's meteorite and these chondrites, they were able to find halite crystals with fluid inclusions and identify carbon-based molecules that had been preserved from the radiation effects from space inside these fluid inclusions. Again, this is not a biological origin. This is just carbon molecules, but it shows that halites are able to help preserve from the degrading effects of even space radiation to preserve a carbon-based molecule. Now, if we want to extend some of these studies, we can use other experiments on the ground, like a solar simulator. And these are ground-based terrestrial experiments that we can use to use a range of different radiation wavelengths. And this helps us to combine different radiation sources. So instead of one kind of radiation, we can have several at a time. Now, I'm not going to go further into this because for that, stay tuned for the talk of Dr. Karen Olson-Francis, which is coming up, and she'll go into more detail about extreme environments and lab simulations. So definitely tune in for that one. The last thing we can do, if we really want to see the combination of all of the individual sources of radiation and microgravity, we need to go directly into space. Space is a selective tool that we use to look at these combinations that we can't reproduce on the ground. And halites, halophiles do have a heritage in spaceflight. Halophiles have been flown on Biopan and on Expose E and Expose R. And these were done with strains that were environmental isolates. They took halite from the environment and put it in space and then measured survival afterwards. So you can see from the image, we stand on the shoulders of giants because there is heritage here for studying halophiles in space. The general idea of these first experiments, because you always have to start somewhere, is to measure survival. Do the cells live? Do they die after exposure to different durations in space? Now, with this important basis of information, we could ask the follow-up question, which is, how are they surviving? We need to be looking at in situ analyses and molecular analyses. We need to get into some more detail. Now, to do that, we also need some more tools. And remember, space is a very difficult environment to work in, and it needs to be used as a selective tool. It's not the answer for everything. And in fact, ground-based studies are often more accessible, less expensive, and gives us access to a greater sample size and analytical power. The true power of doing things in space is the combination of effects that we can't reproduce otherwise on the ground, even if it is with a smaller sample size. So ground-based and space-based experiments should always be worked together to complement each other and not to replace. The experiment that next experiment that will be going up to be looking at these phenomena is going to be ExoCube. So this is a project that is led by Dr. Andreas Elsesser at the Free University in Berlin. And the project ExoCube is to look at in situ analyses of both biosignatures and living microorganisms. Now, designing things for spaceflight is a very difficult process and involves new technologies. In this group, oops. There we are. We work with a large consortium of people that are all going to be providing different kinds of samples, different microorganisms, different biomolecules that are interested to study. We are going to be contributing halophiles and halite to take a look at the effects in space and compare with all of the analyses that we have done on the ground. The ExoCube is set to fly on the ExoBio platform on Bartolomeo Columbus. It's going to be a new exposure platform for the International Space Station. And this is all done with a consortium between ESA, Kaiser Italia, and OHB Systems that are designing this new spaceflight hardware. So as you can see, this is what the hardware rough schematic of what it's going to look like. There's going to be several experiments on the first iteration of the ExoBio experiments. And in these first set of experiments, it'll be ExoCube Chem, ExoCube Bio, the two components of the ExoCube project, OreoCube, which is looking at prebiotic chemistry, and Ice Cold, which is with DLR and Dr. Elke Rabau, who's going to be looking as well at halophiles and other organisms under cold conditions that are closer to icy moons. And then it allows us to compare between different conditions within the same experiment. And of course, there'll be dosimetry to measure the radiation in space. Now, again, designing something like this is very difficult. This is going to be the first time that we're going to be doing in situ analyses on the outside of the International Space Station, including hydrated microbial cultures. And so I want to just take a moment to acknowledge all of the industrial agency academic partners that are working with the scientific team to help develop this hardware. It is a very challenging thing that they are attempting to do, and it can't be done without this kind of teamwork. ExoCube has two components, ExoCube Bio and ExoCube Chem. And it's designed to take a look at multiple samples with dark controls as well as irradiated controls, and to look at cell envelopes and biological membranes as well as microorganisms. So we break it down to the component parts. ExoCube Bio is going to be essentially looking at extant life. How does life, terrestrial life, modern terrestrial life, how does it react exposed to the radiation in space? And for that, there's going to be a series of irradiated samples and non-irradiated samples in a microfluidic system using in situ calorimetry and fluorescence measurements. So this is using a series of LEDs that will help us to rehydrate the cultures in orbit after they've been exposed to a given dose of radiation, rehydrate them, and to see their response and recovery using different fluorescent markers for different cell activity and survival. In our case, will be the project is ExoCube Halo, and that's funded by the INR with participation with CNES. And we'll be flying Halobacterium salinarum inside the halite and fluid inclusions. And the reason we're interested in participating is because it falls along with our philosophy of trying to see inside this closed box, whether on Earth or in space. How do we see what's happening inside halite? So to do this, we are testing an entire series of fluorescent markers with an incredible team of early career researchers and interns that have been working on this, looking at all the fluorescent markers that could be compatible with the extended period of incubation within salt crystals, with the unique physiology of halophiles and halophilic archaea that are compatible at saturating salt conditions, with the radiation conditions in space, and the temperature regime during the entire launch, exposure, and recovery process. The other component of ExoCube is ExoCube Chem, which is focused looking on biosignatures of past life. And for this, again, there'll be a series of irradiated and non-irradiated samples, but in this case, the focus is going to be on cell membrane, cell wall structures, and biosignatures. And again, the idea is to have a magnesium fluoride window, which is going to be allow the solar radiation to pass, expose the different samples from different organisms, and perform in situ Fourier transform infrared spectroscopy in orbit with follow-up samples analysis afterwards. In our case, we will be contributing cell envelopes of halophilic archaea inside halite fluid inclusions for analysis. So with all this together, what are the reasons to be salty? Well, salts give us, in my opinion, one of the great opportunities to look at the limits of life. What are the limits of life? Why are there limits of life? What does that mean for the potential for life on other planets? Salts can be a time capsule of not only potentially living microorganisms, but their biosignatures. How long can those be preserved? How can we detect them? How can we analyze them? This gives us an opportunity to develop new desalination technologies that we use in the lab for our analytical purposes, but that could also have potential for things like spin-off in desalination technologies. And it helps us prepare for things like sample return for Mars and other places where we know there's going to be halites. So with that, I have a lot of people that I need to be thanking who've participated with this, past and present members of the lab, especially these early career researchers who have just been the heart and soul of this project, funding from the French National Agency, the ANR, our wonderful collaborators from the different labs in France and elsewhere that have helped us perform all this work. The collaborative team from the space agencies, industry, and academic partners of ExoCube, and that's all led by Andreas Esser at the Free University, our collaborators in microfluidics in Bordeaux, and all the support that we have received financially from the various agencies. Last but not least, the wonderful memes from Star Trek No Context that have made this presentation so much fun. With that, thank you so much for your attention. I just want to encourage you all, please get in touch with us. We firmly believe that science is participative, it's collaborative, it should be communal. And if we're sending something into space, we want it to be of the greatest good for the greatest number of researchers. So if you think that there's something that we could do differently, better, please reach out to me. I'm happy to chat. I want this to be of the greatest benefit to all of us in the community as we move forward. So thank you very much.